Welcome. Today, we're diving right into the microscopic battlefield where these incredible drugs outsmart deadly viruses. We're going to explore the really elegant and precise mechanisms of antiviral pharmacology, and we'll do it all by following a single patient's journey from diagnosis all the way to treatment.
So let's set the stage. Our story kicks off with JR. He's 26, and he goes to his doctor with what sounds like a pretty standard bug, right? A sore throat, a fever, and he's just really tired. You know, these super common symptoms are actually the starting point for a much deeper pharmacological investigation.
Okay, so the doctor starts running through the usual suspects. A common cold, maybe strep throat, but that specific combination, fever, sore throat, fatigue, that's what doctors call a mononucleosis-like illness. And that particular pattern, well, it raises a bit of a red flag. It pushes the doctor to consider more serious viral culprits like cytomegalovirus or CMV, and, really importantly, HIV.
Now, this is where the story takes a sharp turn. 5 years go by, and JR is back at the clinic. He's now confirmed to be HIV positive, and the virus has done some serious damage. His CD4 count, which is how we measure the strength of the immune system, has just plummeted to 100. Now, to put that into perspective for you, a healthy count is somewhere between 800 and 1200. So at kind of 100, that's a critical state. It means his immune system is severely compromised.
So to fight back, JR is put on a powerful multi-drug cocktail. It's a strategy that we call highly active antiretroviral therapy, or HAART. The whole idea is to attack the virus from multiple angles at the same time. He's also given acyclovir, and that's to treat the painful oral and genital herpes lesions that have popped up. A classic opportunistic infection just taking advantage of his weakened immune system.
So this whole case really sets us up with some critical questions about how these drugs actually pull off their magic at the molecular level. And by the end of this explainer, you're not just going to understand the science behind JR's treatment, you'll really appreciate the incredible ingenuity that goes into modern antiviral therapy. All right, let's get into it. Here's our roadmap. We're going to start with the basics, the conflict between the virus and the host. Then we'll trace the step-by-step invasion plan of HIV. And after that, we'll uncover how each class of drug acts as a kind of saboteur, throwing a wrench into the virus's replication machinery.
First things first. Before we can even talk about the drugs, we have to understand the target. We have to understand the virus itself.
And this definition right here, this is the key to everything. Viruses are the ultimate parasites. Think of them as just biological code, but with none of the hardware to run it. They're what we call obligate intracellular parasites. That just means they absolutely must invade a host cell and hijack its internal machinery to make copies of themselves. Without us, they're basically inert.
And that brings us right to the central challenge of designing antiviral drugs. Since the virus is using our own cellular equipment to work, how in the world do you kill the intruder without killing the cell it's hiding in? Well, the answer is a word you'll hear a lot: selectivity. The best antiviral drugs are like molecular snipers. They're designed to exploit the tiny but crucial differences between viral parts and our own, letting them attack the virus with minimal collateral damage.
Okay, let's zoom in on our main target here, the human immunodeficiency virus, or HIV. You can see its core contains its genetic blueprint, which is in the form of RNA, and that's protected by a protein shell called a capsid. This whole thing is then wrapped in a lipid envelope that it literally stole from a previous host cell. And studded on that envelope are glycoproteins. You can think of these as the molecular keys the virus uses to unlock and get inside our immune cells.
Okay, so now that we know what it looks like, let's talk about what it does. We're going to walk through the entire HIV life cycle, which is basically the playbook our drugs are designed to tear apart.
This diagram, this is our map for the whole invasion. It lays out every single step of the HIV life cycle, from the very first contact and entry into the host cell, through that clever process of reverse transcription and integrating into our DNA, all the way to the final assembly and release of brand new viral particles. And every single one of these steps is a potential chink in the virus's armor, a target for our drugs.
Every one of these steps is absolutely critical for the virus. It's not optional. From recognition and fusion, through reverse transcription and integration, all the way to the final assembly. The virus must complete each step in this exact sequence to replicate successfully. And that linear dependency, well, that's its weakness. If we can block just one of those steps, we can stop the whole process dead in its tracks. So our first point of attack is right at the very beginning of the cycle. We want to prevent the virus from even getting in the door and making a home for itself in our cell's DNA. Let's start with a drug called enfuvirtide.
You can think of it as a bouncer at a club door. See, for HIV to get in, a viral protein called GP41 has to do some pretty complex molecular gymnastics. It changes its shape to pull the virus and our cell together until they fuse. And Fuvertide is designed to stop that from ever happening.
So how does it work? Well, the cause is that GP41 protein has to refold to let the virus in. The mechanism of the drug is that Enfivirtide basically mimics a piece of GP41. It binds right to it and just gums up the works. The technical term is steric hindrance, which is just a fancy way of saying it physically gets in the way. And the consequence? The refolding is blocked, fusion can't happen, and the virus is just stuck outside, unable to get in.
And you know, this strategy of blocking entry isn't just for HIV. Take the influenza virus, for example. Once it gets inside a cell, it has to uncoat itself to release its genetic material. This whole process gets kicked off by protons flowing through a special channel called M2. The drug amantadine works by acting like a cork, physically plugging that M2 channel. No protons get in, the virus can't uncoat, and the infection is stopped before it even really begins.
Okay, now we move to a really critical and frankly kind of terrifying step in the HIV life cycle, integration. This is where the virus literally inserts its own genetic code directly into our DNA. This is the point of no return. Once that happens, the host cell is permanently hijacked. It's now a non-stop virus factory. The drug raltegravir is designed to prevent this permanent corruption of our genetic code.
The viral enzyme that does this is called integrase, and you can think of it as a genetic cut-and-paste tool. First, it does something called 3' processing, where it snips the ends of the viral DNA to get them ready. Then it performs the strand transfer step, which is where it actually pastes that prepared DNA into our chromosome. Raltegravir is a precision tool that works by blocking that second paste step, stopping that final, irreversible connection.
And this image shows exactly how it works. The raltegravir molecule binds directly to that integrase enzyme and just disables it. It stops the paste function, which leaves the viral DNA just floating around in the cell, unable to permanently embed itself into our genome.
The next major target is a really unique process called reverse transcription. This is where the virus turns its RNA genome into DNA, a process that is the absolute hallmark of retroviruses like HIV.
This class of drugs works through pure deception. They're what we call nucleoside analogs, which means they're molecular imposters. They look almost exactly like the natural building blocks of DNA, but they're hiding a fatal flaw. They're missing a tiny chemical group called a 3' hydroxyl. In DNA synthesis, that group is the essential hook for adding the next block. Without it, the chain just comes to a dead end. It's like a faulty Lego brick that stops you from building any higher.
So let's bring this back to JR's regimen and look at zidovudine, also known as AZT. It's an imposter of the DNA building block thymidine. But before it can do its dirty work, it needs to be activated inside the cell. That means it needs to have 3 phosphate groups attached to it. And here's the crucial detail, the part that explains its side effects. This activation is done by our own human enzymes. And that means AZT gets activated in any of our cells that are dividing, not just the ones infected with HIV.
So because our own enzymes switch it on, active AZT can build up in any of our rapidly dividing cells, think about our bone marrow or our gut, and that can lead to toxicity. Once it's activated, it gets incorporated into the growing viral DNA chain, and because it's missing that critical hook, it acts as an obligate chain terminator. It just grinds DNA synthesis to a halt. Now, it is about 100 times better at inhibiting the viral enzyme than our own, but that lack of perfect selectivity is where the trouble can start.
Now, let's compare that to acyclovir, the drug JR was taking for his herpes. It's a guanine analog, and it's also a chain terminator. But the way it gets activated, well, as you can see here, it is brilliantly selective.
The absolute genius of acyclovir is all in its activation. The very first and most important step of its activation is done by an enzyme that is made only by the herpes virus itself. And this viral enzyme is incredibly efficient, about 30 times faster than our own enzymes at this specific job. The result is it's a beautiful piece of pharmacology. The active drug gets trapped and accumulates to concentrations 50 to 100 times higher, but only inside the infected cells. Our uninfected cells are left almost completely untouched.
And this slide brings it all together. This is the key takeaway. Zidovudine is activated by our cellular kinases, so it builds up in all our dividing cells, creating the potential for toxicity. Acyclovir, on the other hand, is a smart drug. It needs the viral kinase to switch it on, so it concentrates its power only where it's needed, inside infected cells.
This is a masterclass in pharmacological selectivity, and it explains perfectly why acyclovir is so much safer.
All right, but what if the virus gets past all those early defenses? What if it actually manages to replicate and make new copies of itself? Well, even then, we have drugs to stop the new virus particles from actually maturing into infectious agents.
And that brings us to the final anti-HIV drug in JR's regimen, ritonavir. And this one targets the final assembly line.
See, during assembly, HIV produces these long, non-functional chains of proteins. You can think of it like an unassembled piece of furniture from a flat pack. To become functional, these big chains have to be precisely cut into individual working parts, and that cutting is done by a viral enzyme called HIV protease. Ritonavir's job is to inhibit those molecular scissors. So the consequence is that new viral particles can still form and leave the cell, but they're completely immature and non-infectious because their internal machinery was never properly assembled. They're just duds.
Okay, let's circle all the way back to our patient, JR, and use everything we've just learned to answer the questions we started with.
So, let's solve the case. This table puts it all together. JR's regimen was a three-pronged attack on HIV. You had zidovudine and lamivudine, both acting as chain terminators to stop DNA synthesis. Then you had ritonavir, the protease inhibitor, making sure any new viruses were duds. And on top of that, he had acyclovir, another chain terminator, but one that was specifically and safely targeting his secondary herpes infection.
And now for the final, really crucial question about toxicity that we've been building up to this whole time. So why is one drug so much safer than the other? The answer is one single powerful word: selectivity. Acyclovir is a targeted weapon. It's activated only by the virus it's designed to kill. Zidovudine is more like a bomb, activated by our own cells, and therefore capable of causing collateral damage. This principle of selective activation, that is the holy grail of antiviral drug design. This last diagram really gives us a bird's-eye view of the entire battlefield. We can see every single point of attack we've talked about. Fusion inhibitors blocking the gate, integrase inhibitors stopping that point of no return, polymerase inhibitors corrupting the blueprints, and protease inhibitors sabotaging the assembly line. It's a perfect illustration of how a multi-pronged pharmacological attack can successfully suppress a really complex virus like HIV. And that leaves us with a final thought. As we keep unraveling these incredibly precise molecular battles, how can we design the next generation of even smarter, even more selective antiviral drugs? The ongoing chess match between viruses and scientists is this amazing story of ever-increasing precision, where every new mechanism we understand opens the door to a new life-saving strategy.